Inside every single one of our cells, there's this incredible ancient defense system at work, constantly on the lookout for genetic invaders. It's a silent battle. But what if we could actually harness that system for ourselves? Well, today, we are going on a journey deep inside the cell to explore the world of gene silencing and a technology that is, quite literally, changing everything.
So let me just start with this question. Imagine you had a switch for the very code of life itself. What if you could find one single troublesome gene, maybe one that's causing a disease, and just flick a switch to turn it off? I know, it sounds like something straight out of science fiction, right? But believe it or not, it's a process that's happening inside living cells every single second.
And nature's answer to that question is a process called RNA interference, or as you'll hear it called, RNAi. The best way to think about it is like a super precise internal security system. It's constantly scanning for and neutralizing very specific genetic threats. And this one biological pathway, it has completely revolutionized how we understand and even treat all kinds of diseases. Okay, so to really understand how RNAi works, we need to meet the key players in this little molecular drama. Let's not think of this like a boring textbook diagram, but more like a cast of characters, where each one has a totally critical role to play in the story of silencing a gene. First up, we have our trigger. This is double-stranded RNA, or dsRNA. It's basically the alarm bell that kicks everything off. Next, we have an enzyme called dicer, which acts like a pair of molecular scissors. Dicer's job creates something called siRNA, and this acts as our targeting guide. Then, this little guide gets loaded into RISC, which is the real silencing machine. And finally, who's the target of this whole operation? That would be the messenger RNA, or mRNA, the genetic message that we need to shut down. All right, let's get into it. It all begins with that trigger. We're going to follow the action step by step, starting right from the moment our cell detects something unusual and kicks its defense system into high gear. So the whole story gets started when a long piece of double-stranded RNA shows up inside the cell. Now, this is a big deal because most of the RNA in our cells is single-stranded. So when the cell sees this dsRNA, it thinks, whoa, what's this? It's a red flag, a potential sign of a virus or some other genetic problem that needs to be dealt with and fast. This is the signal that initiates the entire RNAi pathway. The moment this dsRNA is detected, our molecular scissors, that dicer enzyme, springs into action. It grabs onto that long strand and, with incredible precision, just starts chopping it up. But it's not random shredding, oh no. It cuts it into these perfect, uniform, bite-sized pieces, each about 21 to 23 nucleotides long. These little fragments are now called short interfering RNAs, or siRNAs, and they are the absolute key to what happens next. So the cell has now processed that initial threat signal into a whole set of super-specific targeting guides. The next step? It's time to weaponize them. It's time to build the machine that's actually going to carry out the silencing mission. And that machine is the RNA-induced silencing complex, or as we call it, RISC. This is really the heart of the whole operation. RISC is basically a crew of proteins, including a really important one called argonaute, that all come together to form this programmable silencing engine. But here's the thing. On its own, it's totally useless. It needs instructions. And this is where those siRNAs we just made come into play. One of those little fragments gets loaded into the RISC complex. RISC then unwinds the two strands, gets rid of one, and keeps the other as its guide. So now, armed and ready, the RISC complex starts patrolling the cell, scanning every single messenger RNA it can find, looking for a perfect match to its guide. When it finds that complementary sequence, bam, it latches on. The argonaute enzyme inside RISC then delivers the final blow. It slices the target mRNA into, completely destroying the message. And just like that, the mission is a success. The genetic message is destroyed before it can ever be translated by the cell to make a protein. And if you don't have the protein, well, the gene's function is shut down. It has been silenced. So understanding this beautiful molecular process is one thing. It's amazing. But what makes it truly revolutionary is our ability to actually harness it. See, by creating our own custom-made dsRNA and introducing it into cells, we can essentially hijack this natural pathway to silence almost any gene we choose. So why is that so powerful? Well, it really all comes down to two key words: efficiency and specificity. RNAi is incredibly good at knocking down a gene's expression
And because it all relies on that precise sequence matching, it's like a guided missile. It can find and hit one specific gene out of tens of thousands, leaving all the other ones completely untouched.
And this is where it gets really mind-blowing. The real power of RNAi is in its applications, and they are just staggering. For years, this was just an amazing tool in the research lab, but now it's a whole new class of medicine. The very first RNAi drug was approved back in 2018, and it silences a gene that causes a rare fatal nerve disease, actually stopping it from getting worse. Similar therapies are now being developed for everything from high cholesterol to viral infections and even cancer. And it's even being used in agriculture to design crops that are naturally resistant to pests.
So here we are. We've uncovered one of life's fundamental operating systems. We now literally hold a switch that can, with incredible precision, turn off almost any gene we choose. The therapeutic possibilities are just immense, but so are the ethical questions. As we move from silencing genes in a lab dish to silencing them in people, how do we decide which switches are actually okay to flip?